Good morning, dear children. Today, welcome to our biology class. Today, I am going to discuss about the topics of mechanism of urine formation, ureters, urinary bladder, and urethra. So, these topics we will discuss now. In the previous class, which topic we completed? That is structure of kidney and structure of nephron. So today's our first topic is mechanism of urine formation. So before going to that it is needed to recall the structure of kidney. So structure of kidney and structure of nephron or uriniferous tubule. So structure of nephron has two parts, that is Malpighian body and renal tubule. Malpighian body is consists of cup-shaped structure, that is called Bowman's capsule, and it contains the bunch of blood capillaries, that is called glomerulus. Together of Bowman's capsule and glomerulus is called as Malpighian body or renal capsule, and it is the main place to get the filtration of blood in the glomerulus, means the bunch of blood capillaries in the Bowman's capsule. These are podocytic in nature and they can filter the blood. Next part of the nephron or uriniferous tubule is renal tubule. It is tube-like structures, consist of three parts, that is proximal convoluted tubule, loop of Henle, U-shaped loop of Henle, and distal convoluted tubule. Okay. Now the mechanism of urine formation. It consists of four stages, that is glomerular filtration, tubular reabsorption, tubular secretion, and formation of hypertonic urine. These four steps are involved in the mechanism of urine formation. So what is the first stage? Glomerular filtration. So here blood flows from renal artery to glomerulus through the afferent arteriole. You know that in the glomerulus or Bowman's capsule, two types of blood vessels are there. One is incoming blood vessel, that is called as renal artery, and it is also afferent arteriole. And another blood vessel is there, that is efferent arteriole. This comes outside from the Bowman's capsule. Okay, so here blood flows from renal artery to glomerulus through afferent arteriole, and you observe the diagram in the textbook. You should open your textbook and observe in the diagram also. So here the glomerular filtration in nephron, and try to answer the following questions. So here blood flows from renal artery to glomerulus through the afferent arteriole. This is incoming arteriole. When compared with the efferent arteriole, this blood vessel size is bigger. And blood in glomerulus under the influence of pressure due to the narrowness of efferent arteriole. Efferent arteriole outgoing, existing blood exit outside to the glomerulus. So it is narrow in nature, and that's why it can produce the pressure. That pressure helps to filtration of blood. So what is the reason here to take the filtration of blood? Due to the narrowing of the efferent arteriole that can create the pressure. So that's why here blood is filtered, that is called as ultrafiltration. Next stage, tubular reabsorption. Re means again, absorption means take inside. So here filtrate from glomerular is also called primary urine. So here blood is filtered in the glomerular filtration. So that is called as primary urine. So which is almost contain equal to the blood and in chemical composition except the presence of blood cells. So here what is present? primary urine, here blood cells are not there, and other chemical substances which are needed to our body, they are present. That's why this is called primary urine. So that's why here very very important to absorb the chemical substances which are needed to our body. That's why here reabsorption takes place, that is called tubular reabsorption. So it passes into the proximal convoluted tubule. So in the renal tubule, filtration takes place, that is goes to tubular. Now what is the next part of nephron? That is renal tubule. So this primary urine can goes to renal tubule. So that's why first tubule of the renal tubule is PCT, proximal convoluted tubule. So that's why primary urine goes to or passes into the PCT. Here useful substances in primary urine are reabsorbed into peritubular network. So these all PCT, loop of Henle, DCT is enclosed by the network of blood capillaries, that is peritubular network. So here, did you understand? So here once again, filtration from glomerulus is called as primary urine. So it has some composition of blood except presence of blood cells. So when primary urine passes through the proximal convoluted tubule, the useful substances in the primary urine are reabsorbed. So what are that useful substances? Glucose, amino acids, vitamins like C, K, calcium, sodium, chlorine, and 75% of water. These are needed to our body. So that's why these are reabsorbed. So here are useful substances in primary urine are reabsorbed into peritubular network. Next stage, tubular secretion. After reabsorption in PCT, the urine travels through loop of Henle. What is the next part of renal tubule after PCT? Loop of Henle and DCT. So primary urine after reabsorption, it enters or it passes through the loop of Henle and DCT. Here other wastes like extra salts, sodium, potassium, chlorine, hydrogen are secreted into the DCT from peritubular capillaries. Here other wastes are secreted into next part of the renal tubule. It occurs mostly in the DCT. So this maintains proper concentration of pH of urine. So this secretion of waste materials can helps to proper maintenance of pH to the urine. So smaller amount of secretions occurs in PCT. This is the process occurs in tubular secretion. Fourth stage, formation of hypertonic urine. So in the tubular secretion is active secretion of waste parts by blood capillaries into urinary tubule. They are removal of waste substances like these secretions helps to removal of waste substances like urea, uric acid, creatinine, and ions like sodium, potassium, hydrogen, etc. This helps to maintain the concentration of and pH of urine. Okay, so next one is formation of hypertonic urine. Here 75% of water in nephric filtrate is reabsorbed. So if this 75% of water is excreted, it is very dangerous to our body. So that's why this 75% of water in the nephric filtrate, what happens? Reabsorbed. How much water is passed out? Only 10% of water passed out. So by which process? By the osmosis, where it can passed out in the loop of Henle. So further concentration of urine occurs in the collecting tubule in the presence of hormone vasopressin. So here, if concentration of urine takes place, if every day we can drink nearly 3 to 4 liters of water. If after the filtration of 10% of water is passed out, it is dangerous to our body. It is changes into diluted. So if diluted urine may be causes to the frequently you have the urination sensation. So to prevent that, here one hormone helps to get the urine concentration. So that is vasopressin. If any person has the deficiency of vasopressin, what happened? That person can having the disease frequently urination. That is called as diabetes insipidus. That disease is called diabetes insipidus. You see here, vasopressin. What is its importance? Seeked when concentrated urine is to be excreted. So this vasopressin helps to excretion of concentrated urine. If absence of vasopressin hormone, what happened? Urine is in the dilute form, dilute urine. So maintenance, osmoregulation of blood fluids, deficiency of this hormone causes diabetes insipidus. So these points, these notes will note down in our notebook also. These are the important points already I am giving. So what is the importance of vasopressin? Vasopressin causes concentration of urine. So what is the reason to cause the disease? Diabetes insipidus, deficiency of vasopressin causes diabetes insipidus. This is the formation of urine. Okay, so what is the next topic of today? Ureter. You recall the structure of excretory system. Kidney, pair of ureters, urinary bladder, urethra. These are the parts of urinary system. So kidney and formation of urine, structure of nephron finished. So what is the next part of excretory system of that is ureters. So this entire process is given in the diagram. So glomerular filtration and glomerular tubular reabsorption, tubular secretion. So where they can take place, you see in the diagram also. So next part is ureters. Ureters are from the two kidneys, two tube-like structures are rises. So that's why ureters are paired. And structure is, these are whitish, narrow, distensible and muscular tubules. The length of the ureters is 30 centimeters. And each ureter arises from the hilus of the kidney. Where it goes, it can opens into the urinary bladder. What is its function? This ureter carries urine from the kidney to the urinary bladder. So what is the function of ureters? It carries the urine from kidney to urinary bladder. So the movement of urine in the ureter is through the peristalsis. Peristalsis means movement like a waves. What is the reason to cause the waves? The concentration and relaxation of the tube-like these ureters. So this decreasing the size, that is causes to the peristalis movement. Next part, urinary bladder. It is a medium in size, shape is pear shape and distensible sac-like structure. Where is it present? It located pelvic region of the abdomen. So it has the storage capacity 300 to 800 ml. So when this urinary bladder filled with urine, we have the sensation of urination. Next part, urethra. It is the tube-like structure. It can passes the opening of urinary bladder which are generated by sphincters. So that is 4 cm long in the females and 20 centimeters long in the males. That is called as vestibule. So this is size of the urethra is different in male and female.